Good morning. Can you guys hear me? Hello? Hello? Can you guys hear me?
Yes.
Okay, fine. Are you able to see the slides? Are the slides visible from your end?
Yes.
Okay, fine. So this morning we are going to discuss three important genuses, that's Haemophilus, Bordetella, and Brucella. They're quite a unique group of bacterial agents because they are classified as what we call Coccobacilli. Coccobacilli are basically just very short gram-negative, very short rods. Okay, so all of them are gram-negative, and that's why we're going to discuss them as a group. So start with the genus Haemophilus. So Haemophilus are basically gram-negative Coccobacilli, and they're usually found as colonizers or normal flora in the upper respiratory tract. Uh, so in terms of their growth, they require two very important accessory growth factors, that's hemine and nicotinamide adenine dinucleotide, that's the NAD, which we call it the X factor. So X factor is usually provided, the V factor, so sorry, the X factor is
Excuse
usually
me.
provided by, yes,
Hello, excuse me.
yes, please.
Could you kindly raise your voice a bit?
Okay, I'll try. So I was saying that the species within the genus Haemophilus are usually found as colonizers of the upper respiratory tract. So you find them either within the mouth and the oropharynx. And in terms of their growth and proliferation, they require two important accessory growth factors. That's the X factor and the V factor. The X factor is usually provided by hemine, so that's within the RBCs, while the V factor is very essential when it comes to oxidation reduction processes of the cell. They are also included in what we call the HAFEC group of pathogens. So how pathogens are classified based on their pathogenesis. So they are grouped as HAFEC. In terms of their clinical, in terms of their characteristics, so I talked of them being quite small, and they are described as gram-negative coccobacilli. They are also microaerophilic because they require an additional 5 to like 10% of CO2 to enhance their growth. They are also oxidase positive because they produce cytochrome oxidase enzyme, and you find that some species are also catalase positive. In terms of motility and production of spores, they are non-spore formers, and they're also non-motor because they do not produce flagella. So the common species that are important medically are three. We have the Haemophilus influenzae, Haemophilus ducreyi, and Haemophilus parainfluenzae. So for this lecture, we're going to focus just on Haemophilus influenzae and Haemophilus ducreyi. And these three species are usually distinguished based on the growth accessory factors, that's the X and the V factor. So you find that some species would require either both factors, others will just require the X factor, and the other species may require the V factor. So that's how they are distinguished. So let's start by discussing Haemophilus influenzae. So like I said, majority of the species within the genus Haemophilus are found as normal flora within the upper respiratory tract. So, and for the Haemophilus influenzae, they are usually classified into two major groups. That's the typable and the non-typable strains. So the typable strains, they usually have, they are capsulated. So they have a capsular antigen. And that capsular antigen is divided into like six different groups, okay? Because the six groups are quite unique. They're quite specific for each specific antigen. So we have them designated as A to F. However, it's only Group B that is associated with severe invasive disease. The other groups are basically just colonizers, just like the non-typable strains, which don't have the capsules. So for the Haemophilus species, they require both uh, X and the V accessory growth factors. Unlike the type B strains, the non-typable strains usually cause localized infection, and there's something else I needed to mention. You find that infections due to Haemophilus influenzae are less frequent because of course, the presence of or the use of Haemophilus influenzae type B vaccine, okay, which is usually given to children. So that has greatly reduced the infection rate of uh, infections infection rate due to Haemophilus influenzae. So among the virulence factors, just before I go on, can you guys hear me now? Am I audible enough? Hello?
Yes.
Okay,
Yes.
fine. So among the virulence factors that are associated with Haemophilus influenzae, we have the presence of the capsule, which I mentioned, which is very key in terms of being antiphagocytic, and it's usually highly associated with the virulence of Haemophilus influenzae. So the capsule is quite unique because it's made up of polyribosyl ribitol phosphate, or PRP, designated as PRP. Apart from that, it also has the lipopolysaccharide, which is endotoxic. So it has produces some endotoxic activities, and we have some factors that are very key in terms of facilitating attachment to the host, because remember that attachment to the host has to create that relationship between the pathogen and the host for it to be able to cause the infection. Without that attachment, then of course infection will not take place. So it produces a number of adenosines that allows it to attach to the epithelial cells within the respiratory tract. So we have the IgA protease also that allows for that kind of attachment or relationship, and also the neuramidase enzyme. In terms of clinical manifestations, so Haemophilus influenzae usually causes infection mostly in children between the age of 2 months to 5 years, and transmission is usually person to person. Okay, so like I mentioned, you find that some species of Haemophilus are normal flora within the upper respiratory tract. So they can also cause endogenous infections within an individual. But of course, that is mostly dependent on the immune status of a person. So some of the conditions or the manifestations that are associated with Haemophilus influenza, we have pyrogenic meningitis. We also have epiglottitis, which is a condition where you find that the epiglottis, epiglottis becomes inflamed and then it swells. And of course, when that happens, sometimes it closes off the airway. Okay. Then we have you may also have cellulitis, which is accompanied by bacteremia, septic arthritis, and etc. For the uncapsulated or the non-typeable H. influenzae, I said it mostly causes localized infections like otitis media, conjunctivitis, sinusitis, and sometimes pneumonia. Okay, so you have to differentiate infections caused by the typeable strains of H. influenzae and the non-typeable strains of H. influenzae. So for lab diagnosis of H. influenzae, of course, when you're performing the laboratory diagnosis of H. influenza, you have to take into consideration where the infection is occurring, whether it's in the meningeal CNS system or in the respiratory tract. So that will now prompt what you're going to, which specimen you're going to collect. So for CNS infection, you collect CSF. In case of bacteremia, you get blood for culture, and you can also collect sputum. The first procedure is usually to gram stain the specimen. So here you'd be interested in looking at gram-negative coccobacilli in your specimen. Then, since this is not definitive, then you may need to progress to culture. And in culture, we use chocolate blood agar because this is what is going to give you you remember we talked of the need of X and the V factors. So that is usually expressed in chocolate blood agar. And here, because the organisms are microaerophilic, you need to grow them in like 5 to 20% CO2 at around 36 to 37 degrees Celsius for 18 to 24 hours. So sometimes you may, because they are quite, they are slow growers, you may need to hold this for like 72 hours, okay, before you discard the specimen or the cultures, because they are known to be slow growers. And they're very fragile because they die quite very fast, especially in extreme temperature conditions. And also, so when the temperature conditions are high or even quite low, they die quite fast. And the other thing you also need to consider is that when you're collecting a sample that requires transportation, you need a transport media, because in case of delay, then you might lose the organism because they die quite fast. So on culture, after the growth, you'll be able to see pinpoint colonies, mucoid colonies. They're quite small, okay, but mucoid because of course of the presence of them of the capsule. So further identification, you would require to demonstrate the requirement of the X and the V factors. We say that H. influenza requires both factors. So when you're doing, when you're demonstrating this, what we usually do in a routine diagnostic lab is you get discs that are impregnated with both X and the V factors, and then you put in your culture media where you have inoculated the organism. So when you incubate, after incubation, this is what you'll be able to see. You see growth around the discs that have both X and the V and the V factor. Okay, so that's a clear demonstration of H influenza. And apart from that, you can test for presence of or production of cytochromophilia enzyme, which we say they are positive, and also the catalase enzyme, which we say they are positive for that. For epidemiological purpose, we usually encourage serotyping um, because with this you may be able to establish whether an isolate belongs to any of the six serotypes we talked about. So if you are interested in type B, type A, or so that with that you'll have to serotype the isolate. So I've talked about this. So you note the growth around the discs with the X and V. So that's a clear indication of the need of the X and the V factor. In terms of treatment, Haemophilus influenzae has been known to produce the beta-lactamase enzyme, and this, of course, has reduced their susceptibility to the beta-lactam antibiotic. So in this case, you can use the combination of amoxicillin plus clavulanic acid, which we know clavulanic acid is a beta-lactamase inhibitor. So that combination has been shown to work, but you can also go for third generation cephalosporins like ceftriaxone and cefotaxib, which are also important, okay, in terms of treatment. But always be guided by an AST profile, because you may find that some strains are also resistant to the ceftriaxone or even the amoxiclav. Okay, so always be guided by an AST profile. In terms of prevention, I say that we have a vaccine that mainly targets serotype B, which is usually given as a pentavalent together with diphtheria, pertussis, hepatitis B, and tetanus. So that is usually given to children, and that's why I said infections due to Haemophilus influenzae have reduced gradually, and this is because of the availability of the vaccine. So sometimes when you have individuals who are infected in Haemophilus influenzae, especially in a household, they are usually given rifampin, which is a prophylaxis, okay, to, of course, household contacts. This is for index cases, okay. So this is just given to contact as a prophylaxis. So we move on to the next organism, which is Haemophilus ducreyi. So Haemophilus ducreyi is Coccobacilli. However, it is not a normal flora, just like Haemophilus influenzae, where we said it's a normal flora of the upper respiratory tract, but Haemophilus ducreyi is not a normal flora. In terms of distinguishing it with Haemophilus influenzae, it only requires factor X for growth, and this organism has been associated with chancroid, which is a sexually transmitted disease. So the fact that it is not a normal flora, its presence is usually an indicative of an infection. So the clinical findings of Haemophilus influenzae, I said it causes chancroid, which is a sexually transmitted infection. And chancroid is usually very characterized by painful sores within the genitalia. And sometimes the inflammation can cause regional lymphadenitis, so you'll have swelling within um, uh, the lymph nodes of the groin. So not the chancroid ulcerations, which are basically the ulcers within the genital tract. So not that Haemophilus ducreyi causes genital ulcers. It doesn't cause um, secretions, okay, but ulceration. And of course, sometimes you get regional lymphadenitis when you get pyogenic inflammation. So in terms of lab diagnosis, again, the specimen collection will depend on the site. And for us, since it's a genital ulcer, we'll just get the exudate from the chancroid. And like I said, since they die quite fast, you need a transport media. So put it in Army's transport media. And first thing you'll have to do is gram stain the specimen and check out for gram-negative rods or coccobacilli. Incubation, same as what we discussed for Haemophilus influenzae, same as the biochemical test. So after culture, look out for the pinpoint colonies and then perform oxidescent and catalase test. So in terms of differentiating it with the other Haemophilus species, demonstrate for the ability to grow with the, around strips where you have the X factor. So like for that slide, if you see the strips where you have the X factor, you see growth around those strips, okay? And that's a clear indication of Haemophilus influenzae. No, Ducreyi. For parainfluenza, it usually requires just the V factor. So you will see the opposite. You have growth around the V factor and also X and V. So that's for parainfluenza. But for Ducreyi, you only see growth where you have the X factor. In terms of treatment of Haemophilus ducreyi, you can use azithromycin, also ceftriaxone or ciprofloxacin, but again, be guided by the AST profile of the pathogen. Do you have any questions on Haemophilus before we move on to Bordetella?
No question.
Sorry?
I think there are questions on the chat.
Yeah, that's what I'm trying to see. Repeat on the AST, please. AST for which one? Okay, I'll repeat for both. Someone is complaining I'm going too fast. Sorry about that. I'll try and slow down in the next slides. Does Ducreyi also release beta-lactamase enzyme? We can't write the notes even. I'll share the notes, so don't worry about that. I'll share the presentations. So don't worry about writing. I'll share that. Is that okay? So just take note of what you probably don't see highlighted on the slide. So I'll repeat the. I think I'll go back to Haemophilus influenzae in terms of treatment. Was it treatment? Yes. So I said for treatment of Haemophilus influenzae, because they produce the beta-lactamase enzyme. Okay, so they are mostly resistant to the beta-lactam antibiotics. So in this case, you can use a combination therapy where you combine amoxicillin plus clavulanic acid, because we know clavulanic acid is a beta-lactamase inhibitor. Okay, so it will inhibit the production of beta-lactamase enzyme. So it's been shown to work. Alternatively, you can also go for the third generation cephalosporins. That will also work in this case. But like I said, because these organisms keep evolving from time to time, such that the resistance may not only be because of production of a beta-lactamase enzyme, it could also because of binding protein, then you may need to just rely on antimicrobial susceptibility test for the right treatment to give to your patient. Okay, so take note of that. And then in terms of Ducrey, it's the same thing. So you treat with azithromycin, ceftriaxone, cipro, and erythromycin. Someone was asking if Ducrey also produces beta-lactamase enzyme. Yes, it does. But remember, Ducrey being a pathogen that causes the genital tract infection, so you just use azithromycin or ceftriaxone for treatment. Okay, but also monitor it in case of resistance to some of these agents. Okay, so I'll move on to the next, which is Bordetella. So the genus Bordetella, so we have three important species. That's Bordetella pertussis, Bordetella parapertussis, and Bordetella bronchoseptica. So pertussis and parapertussis are known to cause what we describe as whooping cough or pertussis in humans. But bronchoseptica causes usually respiratory diseases in animals and occasionally in humans. Okay, so the emphasis here will be more on Bordetella pertussis and maybe parapertussis, which are known to cause pertussis in humans. So in terms of their characteristics, they are also gram-negative coccobacilli. I changed that. Sorry about that. Coccobacilli, and they're also encapsulated, so they have a capsule surrounding the cell wall, and they're strict aerobes. Okay, so you find them growing in environments where you have plenty of oxygen. They're also catalase positive and they're non-spore formers. So these are the key characteristics of the organisms of this genus. In terms of virulence factors of Bordetella, we have three main ones. We have presence of pili or fibri, which are known to adhere to the epithelial cells of the trachea and the bronchi. And then they also produce filamentous hemagglutinin, which also enables addition to the affiliated cells of the apparatus. Because remember, what I said about Haemophilus influenzae also, the respiratory tract is usually protected a lot, especially by cilia and also the mucous membrane. Okay. However, when you have organism producing adenosine, either attached to the mucosal membrane or even to the cilia, then that allows for infections to occur. There's someone who's not muted. I don't know who. Thank you. So the filamentous hemagglutinin usually now allows that attachment to the cilia, and when you have that attachment to the cilia, it of course disrupts a lot of activities within the respiratory tract. It then produces pertussis toxin when this has been shown to cause lymphocytosis. Apart from that, it also causes tracheal cytotoxin. So the tracheal cytotoxin is what causes damage of the cilia. And when you have damage of the cilia occurring, or that profound disruption of the mucociliary clearance, because we know cilia is supposed to clear any, unwanted agents within the respiratory tract. So when that happens, it only leaves coughing as the only way to clear the debris from the airway. Okay, and that's why you have that uncontrolled coughing, what you call the whooping cough. So we also have production of adenylate cyclase toxin. And adenylate cyclase toxin is a toxin that has been classified within the family of ADP ribosylating bacterial toxins, which are known to cause an increase in CAM. So when you have that increase in CAM, it basically causes paralysis of various phagocytic functions. And apart from that, it might are the slides moving on your end? Sorry. So when you have that paralysis of the phagocytic functions, sometimes it might also modulate the cellular protein synthesis regulation. So that will basically just cause a lot of cellular damage. It also produces the lipo oligosaccharide endotoxin, which we find within the cell wall of gram-negative organisms. So remember, the lipopolysaccharide is either made of the lipo-oligosaccharide or lipopolysaccharide. Okay. Apart from that, they also have the outer membrane proteins, which are also key in terms of attachment, okay, to the host cell. So in terms of the clinical manifestation of Bordetella pertussis, we have three major phases. We have the catarrhal phase, then we have the paroxysmal phase, and the convalescent phase. So in the catarrhal phase, which is usually the initial stage during the infection, it's usually non-specific, and the symptoms usually resemble that of a common cold. So the patient will just present with a headache, fever, and a running nose. Okay, but then when it progresses now to a paroxysmal phase, that's where you have that intense coughing, which we describe as a classical whooping cough, and that's because that intense coughing would cause basically the patient to have loss of breath. And when you have that loss of breath, the lungs would become so empty, such that when you breathe in, you're breathing in, the air gets that impact on the lungs, and you'll have that whooping sound. And at this stage, the patient also would progress to lymphocytosis, and sometimes vomiting and convulsions may result because of the intense coughing. The end phase is what we call a convalescent phase. So this happens when you have that reduction of the coughing. So the intensity of coughing basically reduces as the patient goes back to a normal state. Okay, so that's what we describe as a convalescent phase, and this would happen either because of treatment has taken place or the infection is just reducing. But depending also on the state of the patient, the infection can progress on to other stage where you have a lot of complications, and these complications may range from bronchopneumonia to mental retardation and also encephalitis. So in terms of laboratory diagnosis of pertussis, so you can collect whatever specimens from the upper respiratory tract, including nasopharyngeal swab, aspirate, or even droplets from the cough, where of course just give the patient a plate and they cough directly on that plate. However, remember, pertussis is very contagious. Okay, so it's not something that you just carry out in a normal routine lab. This has to be done in a reference lab also to avoid easy spread or even outbreaks from taking place. So if you gram stain the organism, you see gram-negative coccobacilli, and for culture we have a special media called the boarded gecko selective media, or even the red and lower agar, which is key for selectively identifying or growing Bordetella. Incubation is usually within 12 days at 37 degrees. So they take up to 2 weeks for them to grow. So it's also a slow grower. And that's why majority of the diagnostic labs would not consider culture because of that length of culture. In terms of biochemical tests, always look out for, you can test for presence of production of cytochrome oxidase because they are oxidase positive. So considering the length that the time that culture takes, most people will prefer some tests that are quite fast, like direct fluorescent antibody assays, which are also quite fast, PCR assays, and also serodiagnosis. Okay, so these ones are prompt, and you can get the results within a day, unlike culture where you have to take around for 12 to 14 days to get the results. The good thing with culture is it can enable you to perform antimicrobial susceptibility tests, because with these other tests, you may not be able to perform antimicrobial susceptibility tests. So in terms of treatment of Bordetella, you can use azithromycin, clarithromycin, or erythromycin. We have a vaccine for pertussis, that's the pertussis vaccine that I told you, it's given as a pentavalent with the H influenzae vaccine. And the use of this vaccine has also reduced infections due to Bordetella pertussis and also parapertussis. Okay, so we don't have a lot of this infection because of the use of the vaccine. So do you have any questions on Bordetella? I don't see any questions on the chat box, so I'll move on. So then we have Brucella, the genus Brucella. So Brucella are also gram-negative coccobacilli, and they're regarded as facultative intracellular organisms. So they exist both in intracellular and extracellular environments. Okay, that's why we are saying they are facultative intracellular pathogens. So they exist as both within intracellular and also extracellular environments. They are mostly destroyed by heat course, this is where you apply pasteurization. And when this doesn't happen, then you'll have a sporadic spread of brucellosis, which is usually considered as zoonotic disease. So some of the major pathogens include Brucella abortus, which is found in cows. We have Suis found in swine, Melitensis found in goats and sheep, and also Canis found in dogs. And humans will acquire these pathogens from these specific animals. In terms of transmission, humans usually get infected through three major routes, either when you ingest food or especially milk products that are not pasteurized. So that would include milk, cheese, yogurt, and etc. Or even when you inhale infected particles, droplets from an infected animal. Direct contact of the pathogen through either a broken skin or even the mucus membrane. Okay, so those are the three major routes in which the infection can come. So persons who are usually at risk, we have, of course, the dairy farmers because they come into contact with these animals day in, day out. We also have livestock handlers. The vets also because they come into contact with these animals during treatment. So when they don't wear the right PPEs, then they can also get infected. The employees in abattoirs or slaughterhouses are also at risk because of that contact, and especially the third um, um, route of infection that we talked about is very key for this group of people, of individuals. Lab personnel, especially when they do not handle these specimens aseptically, then they can also either inhale the aerosols or they can also get the infection through broken skin. So if they are not using gloves to handle these specimens, they can easily get the infection. In terms of virulence factor, we have production of the fact that, of course, that we have the presence of lipopolysaccharides, and lipopolysaccharides usually resistant to intracellular killing by the neutrophils, and that's why it's a very key virulence factor. Apart from that, the fact that the pathogen is able to survive within the intracellular um, intracellular environment within the host cell, then that also prevents phagocytic killing because the host cell will not be able to, the host immune cells will not be able to recognize them as foreign organisms. So they'll just be, once they hide within the macrophages, the host cell will just, immune cells will identify this as just macrophages, not noticing that the pathogen has hidden within the macrophages. So in terms of the clinical manifestation, brucellosis usually it usually varies randomly from asymptomatic infections to sometimes very serious disease. So for most part of the infection, uh, of the infection, you find that the person may present with headache. You may also have undulating fever, muscle pain, sweating, especially at night, and anorexia. And these are quite non-specific symptoms. But then when it progresses after some time, it may progress to hepatomegaly. You may also have splenomegaly and lymphadenopathy, which are some of the key common physical findings. If this uh, um, the infection is not treated at this stage, then complications may occur, and the complications may range from arthritis. You may also have inflammation of the spinal cord, and sometimes also endocarditis. Okay, so I'll include those complications there. I realize I didn't include it on that slide. In terms of laboratory diagnosis of brucellosis, so the specimen of interest here would be blood from an infected individual. You can also get an infected tissue or even CSF, but most diagnostic labs would just use blood. So blood, what we usually do with blood is put it in a blood culture bottle. Okay. So once you put it in a blood culture bottle, you incubate it in a backset machine, and then you come and subculture it on a solid media, which is usually blood agar. So on blood agar, you will see presence of non-hemolytic colonies. And of course, the incubation would have taken place between 4 to 6 weeks. So it takes quite a long time for them to also grow on artificial media. And here you also require, because they are microaerophilic, you need to reduce the concentration of oxygen and put in some CO2. After culture, you need to test for the production of cytochrome oxidase and the catalase enzyme, okay, which are also key. So majority of, because of, again, the complications in terms of culture, so majority of diagnostic lab will always rely on serological tests where they detect for Brucella antigen from the patient's blood. So you test for antibodies from this specimen. So remember, again, I also need to emphasize during culture, because Brucella is also very contagious, the isolation and identification has to be done in a biohazard facility. Okay, so that for you to reduce the transmission level to also the laboratory personnel. So this has also to be done in a biohazard facility. So in terms of treatment of brucellosis, you can give combination therapy of gentamicin and doxycycline. Because remember, your treatment should be able to penetrate the macrophages because of that ability of the organism to hide within the macrophages. So they should be able to penetrate the macrophages and get to the bacteria, which is hidden intracellularly within the macrophages. So you can also give rifampicin for a long period like 6 weeks, and alternatively you can combine cotrimoxazole plus gentamicin or streptomycin. Okay, so those are the treatment options for brucellosis. In terms of prevention, basically we do not have a vaccine for brucellosis for humans. We just have one for livestock. And of course that is used in livestock, and that's one of the ways of eradicating the disease in animals. And of course, once it is eradicated in animals, our chances of it now crossing over to humans is quite low. So that is it with the brucellosis. Any questions? Any questions on brucellosis? Okay, do you need me to repeat any or explain any of the slides? Okay, someone is asking the complications for brucella. So I say the complications that may occur due to brucellosis. You can have arthritis. You can also have infections, inflammation of the spinal cord. And sometimes some patients may also present with endocarditis. Okay, so we have endocarditis. Mm, I'm just writing it on the chat box. Huh? So any other question? So I'll share the notes with the. Do you have a class email? Is it the one? No. So I'll share the notes with the with the class in the class report with in the class email. But this is just for BDS. Do we have one for BFAM? Okay, okay, that's fine. So let me just copy this and then I'll address it disappears. I'll address the questions. Did you get the other notes for staphylococcus? Did you have a lecture on staphylococcus? Did I share the notes? Can't remember. So someone is asking what of hepatomegaly? What about hepatomegaly? Is it as a complication of brucellosis? Is that what you're asking? Please clarify whether it's a complication of brucellosis or pertussis. And then can ceftriaxone plus doxycycline be recommended for for brucellosis. We talked about the treatment options I gave. Just go back to that slide. We talked of gentamicin plus doxycycline. You can also give cotrimoxazole plus gentamicin. So you are asking whether ceftriaxone, because ceftriaxone is a cephalosporin, and then with the doxycycline, no. So you just give a combination of either gentamicin plus doxycycline. Because remember, our interest here is to target the organism which is hidden within the macrophages. Okay, so it has to like penetrate the macrophages for it to get into the intracellular environment. So you cannot use ceftriaxone. That did you confirm the other question? The lymphadenopathy, was it for brucellosis or just check? Or is it also a complication for brucellosis? So complications for brucellosis, hepatomegaly, was it hepatomegaly? Hepatomegaly, no. So you might have for this, yes, you might have an inflammation of the spleen and also the liver. Okay, so it can also complicate the liver and the spleen. So yes, that's right. That's the other complication. So include it among the complications I had mentioned. So I think with that, I will end the lecture. Did you confirm? Did you receive the lecture notes for Staphylococcus? So that I put it also on. Yes, no? Or you have the notes? So I'll just share the ones for brucellosis, for Haemophilus, sorry. Okay, I'll share both just now. So with that, I guess we can, I'll end the lecture. In case you have any questions, just always feel free to either send a text or an email. Okay, have a lovely day.